Hello everyone. This video is intended for everyone who wants to learn what a flow cytometer is and how you can analyze data on a flow cytometer. It's one of the best applications of the technique of optics and physics. Um, and this video is not exactly expert guidance or something of that sort, but it's just an introduction to teach you uh, how you could run a fax machine and get the data and how to analyze the basic data out of it using the software called as FLOWJO, which is widely used. Uh, Before I begin, I just want to thank OBS software developers for giving me this opportunity, and I'm using the screen recording software here right now. Okay, let's get started. As you can see, I want to start off with a small presentation just to give those viewers who do not know what a flow cytometer is and what the technique means or what the actual use of the technique is. This video can also be watched by experts who know what a flow cytometer is and who just want to get started on using FLOWJO as a software for data analysis. So right here in front of me is just few cancer cells. I just downloaded these images from the internet. It's one of the images that I like. And the idea is that one would use flow cytometer to look for certain marks. One of the main applications of flow cytometer is to look for certain marks on top of tumor cells or any kind of cell, as a matter of fact. Right here in front of me is another cell, a cancer cell, which has a lot of receptors on top of it. And the idea of using a flow cytometer is I would use an antibody or design an antibody against a receptor of the cell. So let's say, for example, this antibody goes and binds here, and I would be able to view it. So here you can see this is something what I would call an antigen. Anything where an antibody is going to bind is an antigen, or anything that is an epitope or something that the cell, some sort of material that acts as a biological gateway for or a recognition factor could be called as an antigen. So right here is an antigen, simplified diagram. The idea of the flow cytometer is you would first use a primary antibody, and then you would add another secondary antibody which would have a conjugated fluorophore which emits a certain amount of light. And nowadays you can even buy just antibodies, primary antibodies, and you could conjugate your fluorophore, which could just go bind there directly and just save the cost of buying a secondary antibody and also increase specificity, but they are more expensive. Okay, and so in a typical experiment, you would have positive controls, negative controls, and everything. So let's say, for example, we are looking for this antigen in the cells. The question that we are trying to ask is whether this antigen is present or not. So if I have two populations of cells, one that has these antigens and one that doesn't have these antigens, I could make the difference out by just seeing the whole thing. So when I add my primary antibodies in the solution, these primary antibodies would go and bind onto the antigens. And then when I add the secondary antibody, these secondary antibodies would bind to only the primary antibodies, because the specificity is designed in such a way. And in the negative control, what I would do is just in the false positive, I would just use a secondary antibody. So I know that the secondary antibody would only bind to the primary and nowhere else. So I can make out the difference and then subtract the data and then just ignore any non-specific interactions. This is how a flow cytometer looks like. This is the one that I use in my lab, and it's a BD LSR Fortessa. There are many, and these are very complicated machines, but they are some of the most beautiful applications of the field of optics. It's again a reminder of how physics is the mother of all sciences. Um, so all these beautiful images and beautiful lights here right in front of me. So the basic idea of the flow cytometer is that the cells here would travel through the fluidic channel or a filament or a tube, and each of these cells have been labeled accordingly with different colors. The fluorophores, which for example, the one that I showed you earlier, the secondary antibody conjugated with the fluorophore, which looked something yellowish in color. Um, so the flow cytometer basically detects three things. And this is the premise of the whole experiment, the whole idea of the flow cytometer. Three things, that is the forward scattering, the side scattering, and the fluorescence intensity. So what exactly happens here is you get all the data. So once the cell goes in through the tube, the laser shoots whatever light it is, and the filters allow certain wavelengths to pass, certain not to pass. And when these lights hit the cell, they refract the rays, and the detector detects the ray, and it converts it into a digital format, which then gets converted into something of this sort. Uh, there's a lot of mathematics involved here, and I'm just afraid of it. So, more simplified, the cells plus the antibody, and this antibody would have a fluorophore conjugated to it, which emits the light, something of this sort. And as you can see, as it comes in here, the laser detects three things. That's the forward scattering, the side scattering, and the intensity. So what is a forward scatter? The FSC mentioned here is the forward scatter. So when the light hits the cells, um, it's refracted in the same path. Then it would be called, if it's refracted in the same path, it would be called as the forward scatter. And when the light hits the cells, and if it refracts into a different pattern, then it's somewhere on the side. If not on the straight path, then it's called as the side scatter. These are some basic information which I just got from the bosterbio.com website, and you could just read it and then get a better understanding of it. I know that I'm not doing a good job at explaining it, but the idea is the light hits the cells, and the different diffraction patterns are detected by the detectors, and that's what you detect. So what we are going to be doing today is we are going to analyze the data which is from a certain cell line, and we are looking for a certain protein called as protein X, and just seeing if that protein is there or not. So I would get a lot of cells, put them in the suspension, add my antibodies, and if these antibodies conjugate or just adhere to the ligand or the receptor there, then you could detect if this particular protein is there or not. And the results would be somewhat the sort. As you can see here, this graph on the y-axis has the side scatter result, and the x-axis has the forward angle scatter. So based on it, it gives you an idea of what if it's exactly is a live cell or a dead cell. If the cell is dead, then you could expect it to come under the category of debris, and if it's a live cell, it would come somewhere in the center. And eventually the results would somewhat like this. There are different applications, but what we are trying to do is we're just trying to compare if a certain protein is present or not, and the results usually are represented in this format, where it's like the isotype, the one which is the negative control and the positive control. So this basically means that the protein X is present here, I mean, and the negative control is here. So this is where we just put in the secondary antibody, and this is where we just put in the primary plus the secondary. So that's what the result is. So let's get started with the data analysis on FLOWJO. Sorry. I have the FLOWJO data, which the facts data, which I did it a few days ago, and it's just a simple array of cells, and I know that all these cells are almost the same. They are different, but they behave. They have the same physical somewhat similar physical characteristics, and hence I'm going to use the same template for all the cells. I'll just drag everything here. So as you can see here, the names are 1L, 2L, 3L, 4L, 5L, 12, 22, 32, 42, 52. Basically what this means is that 1L and 12. 1L is the one where I have added the primary antibody and also the secondary antibody, and 12 is the sample where I have only added the secondary antibody. So if the protein X is present, then I would be able to detect a signal in the 1L, because the primary would bind to my protein, and then when the secondary binds, it would emit a certain signal. And for example, in the 1,2, I would just add the secondary samples, and that should probably not give any signal at all because there is no primary. So before you edit, you have to make sure that you do all the you use your secondary as a control, so that you can negate all the unspecific binding. As you can see here, this is the same cell, but the one here, this has primary plus secondaries, and this has only the secondaries. So I'm going to use this as my base template. So the idea in a flow cytometer is to gate the population. I'm going to use the polygon gate here, and what this means is that now I'm going to specify, and I'm going to tell the machine, I'm going to tell the software, only select these cells here. So every dot here is a cell type. So I'm telling the machine, the software, just select these cells, and I would name them tumor cells, because I know that these are tumor cells. And I'm ignoring, I'm just trying to not include the ones here. These are typically debris, and the ones here are typically doublets and singlets, or triplets, or whatever. Doublets and singlets are like multiple signals when the cells have conjugated or some weird data comes out. So you don't want that. What you want is the one in the center. So as you can see here, this graph is SSC versus the FSC. So it's a side scatter versus the forward scatter. And what I would want to do is I want to copy this same analysis to this one, and also to all the other samples. So I'm going to copy this analysis to every other cell type. Usually you wouldn't be doing it, because for every cell type you have to do a gating on a separate gating based on its control. But I know that all these cells are all behaving almost the same. So now I have these, the same gating has been applied here. So we have 92.2% of tumor cells which are which I think are tumor cells, and the same technique has been the same algorithm has been copied here. So it's got some about 92.6% of tumor cells. So now double clicking on the gate will give you those just the population that you just selected. So as you can see here, tumor cells and tumor cells. So this is the one that we just selected. So once you have done the first gating, So you have excluded a lot of unwanted debris and doublets. The second step would be now you have to gate just the single cells. So we are now telling the software, the FLOWJO software, to just select single cells. So for that, I would change the axis of the graph and view it in the forward scattering, the width FSCWA and the FSCA here, A, sorry, and FSCW. So yeah, so what I'm seeing here is FSCW versus FSCA. This further removes the doublets and the debris. So as now you can see that most of my cells are concentrated here. I would just, the strongest signal is here, that means. So I would just go ahead and select this certain population and name it as single cell 1. So all this is just part of refining the process. So the editing I'm going to copy to all the other groups, and I'll just select, double click on the data, and now I have a further refined population of cells which is of good quality. Now to further refine it, I would view them in the site scatter. So here, so as you can see here, when I click on the axes, I should have told it before, I apologize, you could view the cells in different channels. So forward scattering channel, the side scatter channels, the APC channels, the FITC, the PERCP, depending on the dye you use. So forward scattering and side scattering is, you don't need a dye for that. But all these others are basically dyes which are detected in a particular channel. So let's refine our population further. Now I'm going to select only the ones which are here because I think those are the single cells. Oh, sorry, I should have done it here in the secondary only sample. I'm going to name it single cells 2. So what I'm doing is I'm ignoring these parts, and because I think they are doublets and they're not perfectly cells. And I would copy the analysis to the whole group here. So now I have just single cells, just the cells that I need. So what I did was, when I did the experiment, towards the end, I did something called as a dead cell staining. So when I'm doing the experiments, I want to be pretty sure I'm not including dead cells in my data. So I have stained these cells with propylium iodine, the PI stain, which can be detected in the channel, the PERCP. So what you see here right now in front of me is these parts are all dead cells and these are live cells. So the cells which have incorporated the PI stain are usually the dead cells, and hence they are detected in this range. So I'm going to ignore them and just use this, these as live cells. So I'm telling the software, just consider these cells as live cells. And I'm going to copy the analysis to the group. So now I have a further refined set of data. So what I'm going to do now is make a histogram. So what I did here is I'm viewing the x-axis in the APC channel. So uh, the APC channel detects the dye, and the histogram is a representation of the count or the signal here. So to view the comparison, I'm going to compare 1L and 1,2. So I'm going to drag this here. And as you can see, what this data tells me is that the first population of cells that I have, there is no protein. That protein which I'm looking for is not there, and hence my primary data, that is the blue, looks the same as the red. And as we go search for more examples, sorry, I should have copied the analysis. So let's compare the cell 2. So this is cell number 2 with the primary, and cell number 2 with only the secondaries. And we're going to view them in as a histogram. In the APC channel. Pardon me guys, this data set doesn't look that good. Let's search for another data set here. Let's take the cell number 3. So cell number 3 here, sorry, with the secondary. So as you can see here, this is cell number 3 with the primary and the secondary and only the secondary. And we're going to view the histogram. The APC channel. So even these cells do not have that protein. Let's take the example of the 5th cell. So this is a perfect example of how this experiment works. So as you can see here, the cell number 5, the primary and the secondary show, so sorry, the blue is the secondary and the red is the primary here. So the red moved quite here because this means that the cell has that protein, and the blue here means the cell doesn't have that protein. So it's an example of how you can use the fax machine to look for certain proteins where you have designed an antibody for. The job that I did here wasn't that clear, but to give you a understanding, I just want to rephrase whatever I said and into a more simpler form. So whenever you have to look for a certain marker on a cell, what you would do is you would look for an antibody that is complementary to it, and then once you have the antibody, you could either conjugate a fluorophore onto that antibody, or you could use a secondary antibody which is complementary to the primary antibody, and that could give you a signal. So depending on the primary plus secondary and the only secondary sample, you could make a difference of if that protein is there or not. For further doubts, you could email me on the email ID mentioned below, and I would be more than free to explain you how to run a FLOWJO, and whatever facts data you have, I could just tell you how to go about it. Before you go ahead and ask your PI or your guide or any postdoc, you could always feel free to just ask me. I could just give you a heads up of what all questions to prepare and what all to expect from your senior when they teach you how to analyze the data. I hope I have done a decent job in making you understand how FLOWJO works. I wasn't just quite sure about the data set that I had, and I was just analyzing it now, and I thought I'd just make a video. So I hope this helps. Likes, comments, and let me know how I can improve my videos. Thank you.